Susan Hertog.
Thanks.
I'm wondering, because no one has spoken about drug resistance and how the tumors mutate in order to combat the newest form of drugs.
Okay.
And so what happens then, and what is the window?
So it's interesting. I think every one of the speakers indirectly references. I think we'll start with Jose because I think you spoke directly to this and you can expand a little. Maybe we'll go right down the line.
Yeah, so very briefly, that's a very important area of research because it's a huge clinical problem. Patients that benefit from a therapy and then they stop responding at some point. What we are doing now, when we have the capacity for the first time, is to, for those tumors that stopped responding, say to Herceptin or to a PI3 kinase inhibitor or to lapatinib or to any other therapy, what we are embarking our efforts in is in going back, obtaining a rebiopsy of the tumor so we can take a sample of the tumor that has stopped responding. And then we can sequence the tumor. We can analyze it very carefully.
Tell everybody what sequence means. I'll let you get away with it once, but this time you have to tell them.
So sequence is we have now the capacity to look at the whole genome of a tumor. So, the tumor has multiple genes, and what we know is that there are mutations that should be all right.
Yes.
Mutations that occur that are responsible for the behavior of a given tumor, and they're responsible for the tumor not responding any longer to the prior therapy. So the concept is that you biopsy the tumor, you sequence, that is, you look at the mutations that are present in the DNA, and you identify the genes that are responsible for the resistance. And that opens you the door to do new therapies. This is something that, interestingly, has been done very successfully by our lung cancer colleagues. They do this routinely, and they do a great job at doing that. So I think that in breast cancer, and I believe, and this is something that maybe, Cliff, you want to talk about, there is a BRCRF initiative precisely to address this topic.
Yes, we'll be talking a little bit about this later in the morning. But, one of the things to make it is maybe overly simple, and I think there are some people in the audience who might want to speak to this, is a basic belief that the mechanism of resistance is the development of a change in the genetic material of the cancer so that it no longer is sensitive to a particular treatment. And that raises really complicated questions about whether that was there from the beginning and is what grows after you kill the sensitive ones, or whether it's something that truly emerges under the direct pressure of the treatment that's effective. And also the timing of it, and also the other factors, including how the body metabolizes and clears drugs. These are all, and I'm being somewhat superficial about that, but these are all complexities that we want to address. I saw that there were probably other answers to this question. Maybe, Kathy?
Well, so there's a way that we can get that information that Jose has used in some trials, and we are trying to get that information with one of the projects that the BCRF is supporting. That many women newly diagnosed with breast cancer will receive all or a portion of their chemotherapy or their Herceptin or the therapies that will affect the whole body before they have surgery. And we technically call that neoadjuvant therapy because the neo part just comes first. Some of those patients will have complete disappearance of the tumor. So even at the time of surgery, the pathologist takes lots of slices, they look very hard, and they can't find any active cancer. We know those patients have an excellent prognosis, but many of those women don't have complete disappearance. Their tumor is still there. That's an excellent setting to try to understand resistance, because you've just proven this tumor, at least a portion of it, was resistant to this therapy. The woman had a biopsy at the time of her diagnosis, and she just had surgery. So you have those paired samples, and when you know that they are at higher risk, you can use that population to look at novel therapy. So one of our BCRF-supported trials is doing exactly that in women with very aggressive disease to try to both improve their outcome and then use the study of those tumors to figure out why their outcome was poor, how the new therapy works, and who it works in.
So this relates in part to your question about clinical trials participation. I have to highlight this. This requires brave patients who are willing to allow us to do a biopsy that might not be medically needed clinically for them today, but will be critically useful in learning what's working, what's not. Now, one can imagine a future where those sequential biopsies would matter. You would actually change therapy maybe based upon them. It's not unimaginable. But it requires right now tremendous sacrifice, which we're well aware of from patients. Gabe, you know anything about neoadjuvant therapy?
How do you spell that?
Yeah.
Well,
Yes. So the joke is that Gabe is probably the leader in the development of the routine use of preoperative therapy or neoadjuvant therapy for patients who have curable early-stage breast cancer. And we have friendly, fun-filled debates about the pros and cons of this as a routine approach, but there's no doubt at all about the fact that it is a tremendous platform, as you just heard, for drug development.
so in reality, over the past 40 years, much of our work has been focused on drug resistance.
Right.
Drugs that are familiar to you, such as Taxotere and Taxol, were developed on the basis of patients whose tumors have become resistant to adriamycin, for instance. Much of the newer drugs, Xeloda, for instance, Exempra, Halaven, all of these were developed in patients who had tumors that had already demonstrated resistance to our frontline agents. So this has been an ongoing effort. What has changed over the past 10, 15 years is our ability to understand the mechanisms of resistance and seek out the information that will actually guide us, develop drugs or drug combinations that will focus on that specific mechanism of resistance, as was mentioned by Jose a moment ago. And I think that is a major change in how we work on drug resistance. We now have the ability to identify molecular changes in resistant tumors and develop from libraries of chemical compounds, or identify, not develop, but identify from huge libraries of chemical compounds, drugs that might fit best the molecular profile or configuration of the resistant tumor. And I think that has been a major, major advance. Much of that has been done by people in this room and others who are not here today, but with support of the BCRF.
Great, thanks. Jed, you want to say something too, sir?
Yeah, I think that the emergence of drug resistance is one area also where an approach that targets the immune system presents some advantages because the immune system is it's a dynamic living organ, and so it has the potential to respond to new challenges that the cancer cell may throw its way. And this is, I think, why when we see patients who do have a response to immunotherapy, those responses tend to be very durable. We don't see many people relapse after immunotherapy. Our challenge is more getting more people to respond in the first place. And so that's why we believe that combinations of therapies that enhance the immune system with therapies that kill tumor cells is really the most promising future.
Great, thanks.